please.
A very good afternoon to all of you, and I'm really glad present from University of Calcutta, Kolkata, India, to present this talk in this international forum, the first International Epigenetic Congress, and thank you organizers for giving me this wonderful opportunity. Today I will speak on arsenic and cancer from an epigenetic perspective. Arsenic contamination is a global scenario, and you know that about 70 countries across the globe, about 200 million people are at risk. And from that, 90 million people are from India itself. So what are the major causes and why? So one of the major causes is definitely the groundwater. That groundwater is used for the drinking water, cultivation, dietary sources, cooking, daily uses, and all this. Apart from this, there are some occupational exposure as well in different parts of the country. But in India, from where I belong, we can find that there are 7 different states which are highly affected with arsenic, and among them, West Bengal is worst hit. And again, if we see that I have already mentioned that arsenic in groundwater is highly contaminated, and that groundwater is used for the irrigation purposes. And among this arsenic affected states, Uttar Pradesh and West Bengal are rice producing countries. This rice is a staple food in our area, and the rural people eat almost thrice a day based on rice. And we have found that this rice contains high amount of arsenic. We have extensively studied in the areas of arsenic endemic areas. We have collected the cooked rice samples from there, that is what we consume, and assessed urinary arsenic of the participants, and then also identified the micronuclei. That's one of the biomarker that we can assess from urinary epithelial cell, give us an indication of genetic damage. And the work has been published in scientific reports where we have shown that arsenic contamination beyond 200 microgram per kg causes genetic damage. And another interesting study in this regard has been conducted is that what we should do, what the entire global population or people or scientists are working for, we are trying to reduce the arsenic threshold level. The present World Health recommendation is 10 microgram per liter or 10 ppb, but still scientific community is trying to decrease it further. We have taken a wonderful 2-wave cross-sectional study. That is, we have taken a same number of population in 2 different time period with a 5-year span and studied arsenic in water as well in their biological sample, that is urine. That we have found along with the government initiation when we are providing them safe drinking water that is below 50 microgram per liter. That time 50 microgram per liter was used as a standard in India and long back, and only after a prolonged time, then only now the standard is 10 microgram per liter. So even if at 5-fold higher, that is 50 microgram per liter, we found that when arsenic content in drinking water is down, the urinary concentration is also down. But this does not solve the entire problem. Arsenic is a system toxicant. It affects a number of systems, a number of internal organs, leading to cancer outcome. And that's are not reversed back. So why? I'm just giving you a simple example from my own studies. We have found that these 2 brothers, they are from the same family, one with several skin lesions and cancer, while the other do not have any lesion despite their environmental exposure is same. So we thought that definitely there are the genetic factors. So And in the field, we found that is only 15 to 20% of individuals, they are showing arsenic-induced skin lesion. Rest of the individuals remain asymptomatic. I want to show this picture that's showing one lady, the mother of these two girls. She is asymptomatic, but the little girl is showing symptoms at so early age. So what are the different modes of mechanism? Is only genetic or epigenetic? That is the main area of research. So we took a very simple strategy. We studied the population. As I have already mentioned, only 15 to 20% show skin lesion, but 70 to 75% individuals they are asymptomatic in terms of arsenicosis symptoms. So what we have to do, we taken the blood samples and the tissues of the affected patients, and then we did the DNA, RNA, protein isolations, and also studied it by sequencing or microarray or at the proteomics level, and also collected tissues, did the histopathology, identified the type of cancer we are finding in this population. So it's just to mention once more that once arsenic is consumed, so that is the inorganic arsenic. Just once it's inside our body, it goes through a metabolism, and in this metabolism, it produces the organic metabolites, that is the MMA and DMA. Both of these variants remain in different valency states, and class 3 variants are always more toxic than the pentavalent form. And I found that AS3MT is one of the important enzyme that works in this entire pathway and plays a very important role. So you can understand from genetics that if there is a sequence variation in this arsenic 3-methyl transferase gene, then also we can expect some difference in outcome, because this is one of the very important gene. And we have identified one SNP, that is RS9527, which is also observed in Bangladesh population by a genome-wide association studies, and we have correlated its association with urinary MMA or DMA excretion. More the individuals, which kind of urinary metabolite a patient excretes, that whether it's more the MMA or more the DMA, this is a monomethyl variant or dimethyl variant. Depending on this methylation index, the toxicity outcome differs, and that again depends on one of these very important gene. But definitely then you would ask that is this the only gene? No. Definitely the answer is no. There are multiple pathways, and I have already mentioned that arsenic is a system toxicant. As a genotoxicant, it is an epigenetic mutagen as well. So it affects a number of pathways, and we have studied all these different pathways. But today, due to this short limitations of time, I'm not going to describe all these pathways, only highlighting the key genes and mainly focus its epigenetic regulation. As I have already mentioned, that definitely it should not be the only one gene, that is AS3MT, to be worked on. So we have to look at the genomic profile. So we have done a microarray, so to identify the candidate gene approach. So what we did, we took 60 individuals, 20 in each of the group, that is one in the arsenic unexposed group, one those who have skin lesions, and those who do not have skin lesions. So among these three groups, we studied the entire 24,000 genes. Hello, am I audible?
Yeah, we can hear us. You may continue.
Okay, thank you. So we have categorized this 24,000 genes and seen its expression profile in these three groups, and found and identified that which are the genes which are upregulated and downregulated. As I'm mentioning again and again, that only 15 to 20% individuals are symptomatic, rest of the individuals do not show any lesion. So it is very important or crucial to identify that which are the genes that are differ between this. So now to identify from microarray, it comes up a lot of genes which are significantly upregulated and downregulated. So the most crucial thing is to narrow down to a handful number of genes, maybe 5 or 10. So which could be the most important panel genes or the signature identifiers of this arsenicosis. And now this part is going on with some mathematical modeling. So we have already done this microarray and found out important significant genes of this pathway, but now we are trying to further narrow down to identify these candidate genes. So that typical picture that I have shown, that even in the same family, we do not get the similar outcome. So it reminds me again and again, it's not only the gene, it's not only the environment, and there are definitely the gene environment crossed out. And now we all know that epigenetics is a key player, and which goes through the promoter DNA methylation, histone modifications, and mitochondrial DNA methylation as well. So DNA is not enough to decide our density, destiny. Sorry. So what we did to start with epigenetics, we took a entire panel of total H3. It contains 24 genes, 24 candidate genes, and we have seen with our samples to identify that which particular signatures we are getting for the arsenic exposed individual. We found that there are some genes which are typically up and some genes which are typically down. So we found that there are significant upregulations and downregulations, and found that is one significant gene is H3K79 monomethylation, which is a DNA damage docking site. Similarly, we also get important downregulation in H3K36Me3, that is the mismatch repair recognition. And similar way, we also made a panel for H4 and identified some important regulators which are altered in arsenic exposed population. So we tried to find out one after another by its candidate approach, just trying to find out the upregulators and its downstream. So first, coming to H3K79 monomethylation, we have already found that H3K79 monomethylation is going up. We did for the western, and then we tried to identify who is in its upstream. We found that DOT1L is the upstream regulator, and in its downstream, there is 53BP1. This is another gene which serves as a tumor suppressor gene. So in one case, it causes the DNA damage to be high and downregulate the DNA damage repairing gene. So this is a summary that once arsenic causes this epigenetic alteration, it causes gamma H2X upregulation, H3K79Me1 upregulation. There's a DNA damage. It's showing that is a DNA damage is more, and the same way 53BP1 is downregulated. So altogether, double strand DNA break is the ultimate outcome. So it causes DNA to be more damaged. So further detailing, if you are interested, you go through this publication where this work has been given in details. So I'm again reminding you this panel analysis, and the second candidate gene, the second candidate signal is mismatch repair recognition, that is H3K36Me3. And we have found that this is a mismatch repair recognition. So again, so this is very crucial that DNA damage is going high. At the same time, DNA repair mechanism is going down. And we have checked this recognition, damage repair recognition pathway. And here we have taken MLH, MSH2, this entire complex. And you can see here that this promoter hypermethylation causes these genes to be downregulated, and this altogether H3K36Me3 is also altered, and these altogether causes an impairment in recruitment of MMR complex. That is, until and unless MMR complex is recruited, it is difficult that DNA damage will be repaired. So in one case, DNA damage high, and in another case, DNA repair is downregulated. Altogether, persistent DNA damage exists. And this part is also published in Elsevier. As I have mentioned that I have shown you a picture or diagram in the right side of this panel that a mother is affected, and we don't know yet whether the baby will be affected. And we have seen a small little baby, so 2 years old only, she is also showing arsenic induced skin lesion. So how it's going if mother is affected, and mostly you know that the mitochondrial transmission occurs through mother only. So if mother is affected, its mitochondrial genome is altered, then it transfers its mitochondria to all its children. And from there, again, from maternal transmission occurs. And in mitochondrial regulation, there is a crosstalk between nucleus and mitochondria. As arsenic toxicity causes a direct effect on mitochondria, it causes increased level in ROS, that is a reactive oxygen species, goes very high, change in mitochondrial membrane potential. It causes mitochondrial structural alteration, mitochondrial mutation, it causes DNA damage, and also regulate very important regulators like PCG1, NRF1, NRF2 in the nucleus, which cross-regulate or crosstalk with mitochondria through MTFA. So here we took two important genes. One is the D loop and another is ND6, because D loop is a site where replication and transcription starts. Therefore, these and ND6 methylation, alteration in this methylation of ND6 is also reported in number of diseases. Therefore, these two are important area which needs to be observed, that how arsenic alters their methylation pattern. So we have analyzed these genes, their mitochondrial methylation status in each of these cases, and compared with the patient's or individual's urinary arsenic status. So we found a correlation between this mitochondrial methylation and arsenic exposure. So what we find, what we see here, arsenic toxicity causes an impact on mitochondria. It causes a D loop and ND6 mitochondrial DNA methylation, altered its methylation. We observed hypomethylation in both of the cases, which causes higher expression, and thus it shows that arsenic induced mitochondrial epigenetic modifications takes place. So what we have observed? We have observed that it's not only impact on mitochondria in enhancing the toxicity and its regulation, it also causes mitochondrial biogenesis regulatory genes. So we have taken tissue samples as well. So arsenic-induced cancer patients, we did the biopsies, we had the clinicians did the biopsies, we took the tissues, and we have studied the mitochondrial, how mitochondrial copy number is altered. We have found that these important mitochondrial biogenesis regulatory genes are getting upregulated, and it's more distinct in tumor tissues. It's a promoter methylation, and we have found a significant correlation with its promoter methylation. These are the series of experiments that we have done, and analyzed with the promoter methylation, and it shows that arsenic induced mitochondrial biogenesis is one of the important factor for this. So in this mitochondrial biogenesis, this is very important, that how this mitochondrial fission fusion goes on. So this is mitochondrial fission fusion goes on in a dynamic process. So it fusion occurs, then this fused mitochondria again goes through the different fission cycles. So this goes on, and there are some mitochondrial fusion related genes, that is MFN1, MFN2, OPA1, and there are some fission related genes, fission FIS1 and DRP1. And a number of in a number of diseases, including cancer, it has been found that if there is an imbalance in between fission and fusion regulatory genes, so it causes a disease outcome. And we have seen here that this fission fusion is pathway is imbalanced. So here is a summary of this slide. That is a nucleus. You can see there is a hypomethylated promoter of PDC1 alpha, and this is regulating, since it is hypermethylated, more PCG1 alpha, its expression, and it causes NRF1, NRF2 expression to be more, and thus it regulates that increased mitochondrial biogenesis, increased mitochondrial DNA copy number, and it also alters mitochondrial fission fusion. And this work has recently published in Chemosphere. Another important regulator is telomere. As we know, that telomere is an important regulator for maintaining the chromosomal end or the stability. So subtelomeric methylation, we have studied this subtelomeric methylation pattern, and the subtelomeric methylation.
Dear Ms. Pritha, we are closing to our deadline, so we are kindly requesting from you to collect your collection thoughts and slides in few minutes. Is it possible?
Yes, sure.
Thank you very much.
So we have found this. We have already identified that telomere is a very important part, and telomere regulation plays very importantly. And here in epigenetic as well, we found that subtelomeric methylation is altered. And we have found that H4K20 methylation is regulated, and by this is altered, which is due to arsenic exposure, and it helps in the alteration in subtelomeric DNA methylation. Loss of heterochromatinization of telomeric DNA causes, and mainly it is not by telomerase driven, this is by ALT pathway. This work has been published in Science of the Total Environment 2020 only. So this is my lab and all the scholars, and he is Dr. Sandeep Bhattacharjee, the clinical collaborator. I am presenting the work mainly from my first PhD student, Dr. Pritha Bhattacharjee. She is also Pritha, Tamalika Sannal, Ankita Das, and Dr. Manabi Pal. And here are by other scholars. All those are going their PhD. Thank you so much.
Ms. Pritha, thank you very much for your valuable contributions. Actually, we were collected all the questions while you are presenting, so we will align with your schedule half an hour later if you are available.
Yes, sure. Thank you.
Thank you very much.
Evet, değerli misafirlerimiz ve sayın hocalarımız. Oturma